Dear viewers, today we will deal with the topic Krebs cycle. The Krebs cycle or citric acid cycle, also known as tricarboxylic acid cycle. The Krebs cycle is a series of chemical reactions used by all aerobic organisms to generate energy through the oxidation of a state derived from carbohydrates, fats and proteins to carbon dioxide and water. In addition, the cycle provides intermediates used by the cell for synthesizing important biomolecules such as aspartate, glutamate that is used in numerous biochemical reactions for the biosynthesis of compounds including phytol chain of chlorophyll, carotenoids, terpenes and gibberellins. The name of this metabolic pathway is derived from citric acid, a type of tricarboxylic acid that is first consumed and then regenerated by the sequence of reactions that occur during the cycle. In addition, the cycle consumes a state in the form of acyl coenzyme A. Pyruvate, the end product of glycolysis, enters the citric acid cycle after it has been converted to acyl coenzyme A, reduces NAD positive to NADH and produces carbon dioxide. Although pyruvate is technically not a part of the cycle, it does provide the major link between glycolysis and subsequent carbon metabolism. Note that pyruvate is produced in the cytosol while the enzymes of TCA cycle are located in the mitochondria. Thus, for pyruvate to be metabolized by the citric acid cycle, it must first be translocated through the inner membrane. This is accomplished by a pyruvate OH negative antiport carrier, that is, pyruvate is taken by the mitochondria in exchange for hydroxyl ion carried into the intermembrane space. Once inside the matrix, pyruvate is oxidized and decarboxylated by large multi-enzyme complex pyruvate dehydrogenase. The NADH generated by the TCA cycle is fed into the oxidative phosphorylation pathway, and the net result of these two closely linked pathways is the oxidation of nutrients to produce energy in the form of ATP. Eukaryotic cells, the citric acid cycle occurs in the matrix of the mitochondria. Bacteria also use the TCA cycle to generate energy, but since they lack mitochondria, the reaction sequence is performed in the cytosol with the proton gradient for ATP production being across the plasma membrane rather than the inner membrane of the mitochondria. The cycle was first postulated as the pathway of pyruvate oxidation in animal tissues by Hans A. Krebs in 1937, for which he earned Nobel Prize in Medicine in 1954 for this outstanding contribution. The citric acid cycle, also called tricarboxylic acid cycle, because for some years after Krebs postulated the cycle, it was uncertain whether citrate or some other tricarboxylic acid such as isocitrate was the first product formed by reaction of pyruvate and oxaloacetate. An overview of TCA cycle. TCA cycle operates under aerobic conditions only, oxidizes 2-carbon acyl coenzyme A to carbon dioxide, produces reduced coenzymes NADH and FADH2 and 1 ATP directly. Reactions of glycolysis are localized in cytosol and do not require any oxygen, whereas pyruvate dehydrogenase and TCA cycle reactions takes place in mitochondria where oxygen is utilized to generate ATP by oxidative phosphorylation. Reaction of pyruvate dehydrogenase complex. The conversion of pyruvate to acyl coenzyme A catalyzed by highly organized multi-enzyme pyruvate dehydrogenase complex is an oxidative decarboxylation. In the overall reaction, the carboxyl group of pyruvate is lost as carbon dioxide, while the remaining 2 carbons form the acetyl moiety of acetyl coenzyme A. The reaction is highly exergonic and is essentially irreversible in vivo. Pyruvate combines with acetyl coenzyme A in presence of pyruvate dehydrogenase complex gives acetyl coenzyme A and carbon dioxide. NAD positive gets reduced in the reaction to NADH. Pyruvate dehydrogenase complex is an assembly of 3 enzymes, 3 individual enzymes: pyruvate dehydrogenase represented by E1, dihydrolipoyl transacetylase represented by E2, and dihydrolipoyl dehydrogenase represented by E3. The oxidation of pyruvate to acetyl coenzyme A involves the coenzymes thymine, pyrophosphate, lipoic acid, FAD, NAD positive, and coenzyme A, acting in association with E1, E2, and E3 in the presence of pyruvate dehydrogenase complex. The Krebs cycle has 8 steps. Krebs cycle consists of 8 cyclic steps producing an equal number of organic acids. Each step is catalyzed by a specific enzyme. The 8 steps involved are: formation of citrate, step 2, formation of isocitrate, step 3, oxidation of isocitrate to alpha-ketoglutarate and carbon dioxide, step 4, oxidation of alpha-ketoglutarate to succinyl coenzyme A and carbon dioxide, step 5, conversion of succinyl coenzyme A to succinate, oxidation of succinate to fumarate, step 7, hydration of fumarate to malate, then oxidation of malate to oxaloacetate. Now these steps in detail. Formation of citrate. The cycle begins with the condensation of an oxaloacetate and the acetyl group of acetyl coenzyme A to form citrate. This reaction is catalyzed by citrate synthase. However, during this reaction, an intermediate citriol coenzyme A is formed, the hydrolysis of which makes the forward reaction highly exergonic. The large negative energy change associated with the citrate synthase is essential to the operation of the cycle. This is the reaction in which acetyl coenzyme A combines with oxaloacetate in presence of citrate synthase gives citrate. Step 2, formation of isocitrate via cis-aconitate. An isomerization reaction in which water is first removed, dehydration, and then added back, called hydration, moves the hydroxyl group from one carbon atom to its neighbor. The enzyme aconitase catalyzes the reversible transformation of citrate to isocitrate through the intermediary formation of tricarboxylic acid cis-aconitate. This is the reaction in which citrate first undergoes dehydration in presence of aconitase, forms an intermediate cis-aconitate. This intermediate undergoes hydration in presence of the same enzyme aconitase and produces isocitrate. Step 3, oxidation of isocitrate to alpha-ketoglutarate and carbon dioxide. Isocitrate is oxidized and decarboxylated to alpha-ketoglutarate. In the first of 4 oxidation steps in the cycle, the carbon carrying the hydroxyl group is converted to a carbonyl group. The immediate product is unstable, losing carbon dioxide while bound to the enzyme. The oxidative decarboxylation of isocitrate is catalyzed by isocitrate dehydrogenase. This is the reaction in presence of isocitrate dehydrogenase gives alpha-ketoglutarate. In this reaction, NADP positive gets reduced to NADPH plus H positive. Step 4, oxidation of alpha-ketoglutarate to succinyl coenzyme A and carbon dioxide. In next step is another oxidative decarboxylation in which alpha-ketoglutarate is converted to succinyl coenzyme A and carbon dioxide by the action of alpha-ketoglutarate dehydrogenase complex. NAD positive serves as electron acceptor in this reaction. The energy of oxidation of alpha-ketoglutarate is conserved in the formation of thioester bond of succinyl coenzyme A. This is the reaction where alpha-ketoglutarate gives succinyl coenzyme A and carbon dioxide. Again, NAD positive gets dehydrogenated into NADH positive. Step 5. Conversion of succinyl coenzyme A to succinate. Succinyl coenzyme A, like acetyl coenzyme A, has strong negative free energy of hydrolysis of its thioester bond. Delta G is equal to 36 kilojoules per mole. Energy released during the breakage of this bond is used to derive the synthesis of a phosphoanhydride bond in either GTP in animals or ATP in plants, and succinate is formed in the process. During the conversion of succinyl coenzyme A to succinate, as it involves the ATP formation directly linked with the substrate conversion, hence it is referred to as substrate level phosphorylation. The reaction step is catalyzed either by succinyl coenzyme A synthetase or succinic thiokinase. The reaction is as follows, in which succinyl coenzyme A gets converted into succinate and gives GTP in case of animals and ATP in case of plants. Step 6. Oxidation of succinate to fumarate. The succinate formed from the succinyl coenzyme A is oxidized to fumarate by the flavoprotein succinate dehydrogenase. Electrons pass from succinate through the FAD and iron sulfur centers before entering the chain of electron carriers in the mitochondrial membrane. Electron flow from succinate through these carriers to the final electron acceptor. Oxygen is coupled to the synthesis of 2 ATP per pair of electrons. This is the reaction where succinate gets converted into fumarate in presence of succinate dehydrogenase. FAD gets hydrogenated to FADH2. Hydration of fumarate to malate. Addition of water to fumarate places a hydroxyl group next to the carbonyl carbon. Enzyme catalyzing this step is fumarase. This is the reaction where fumarate gets hydrated into malate in presence of fumarase. The last step, oxidation of malate to oxaloacetate. In the last of the 4 oxidation steps in the cycle, the carbon carrying the hydroxyl group is converted to a carbonyl group, regenerating the oxaloacetate needed for the continuity of the cycle. This reaction is catalyzed by malate dehydrogenase. This is the reaction where malate gets converted into oxaloacetate and NAD positive gets hydrogenated into NADH plus H positive. Now the importance of TCA cycle. The TCA cycle produces high energy phosphate, ATP and GTP in case of animals. Number 2, it is the main source of reduced coenzymes NADH and FADH. Number 3, some important intermediates of TCA cycle are used by the cell for synthesizing important biomolecules such as aspartate and glutamate when required. Number 4, acetyl coenzyme A produces compounds for the formation of fatty acids, cutin, aromatic compounds, phytol chain of chlorophyll, carotenoids, terpenes and gibberellins. The Krebs cycle or citric acid cycle, also known as tricarboxylic acid cycle. Krebs cycle is regarded as the central pathway of the cell because the catabolic pathway converge upon it and anabolic pathways diverge upon it. Therefore, it is often called as amphibolic pathway. Anaplerotic reaction. Since TCA cycle intermediates are used for anabolism, their concentration varies according to the needs of the cell. So, the anabolic nature of Krebs cycle has raised the question of how the level of intermediates can be replenished when certain intermediates are removed for anabolic purposes. It was the Korenberg who suggested and proposed the term anaplerotic for these replenishing or gap-filling reactions or mechanisms. Oxaloacetate can be considered as a primary substrate of the TCA cycle. It is replenished from pyruvate by the glyconeogenic enzyme pyruvate carboxylase. This is the reaction where pyruvate plus carbon dioxide ATP and water gives oxaloacetate plus ADP plus inorganic phosphate. Pyruvate carboxylase is activated in presence of acetyl coenzyme A. Pyruvate can also replenish malate. Now the amphibolic nature of TCA cycle. In the aerobic organisms, the citric acid cycle is an amphibolic pathway, one that serves in both catabolic and anabolic processes. Besides its role in oxidative catabolism of carbohydrates, fatty acids, amino acids, the cycle provides precursors for many biosynthetic pathways through the reactions that serve the same purpose in anaerobic ancestors. Alpha-ketoglutarate and oxaloacetate can, for example, serve as precursors of the amino acids aspartate and glutamate by simple transamination. Through aspartate and glutamate, the carbons of oxaloacetate and alpha-ketoglutarate are then used to build other amino acids as well as purines and pyrimidine nucleotides. Oxaloacetate is converted to glucose in glyconeogenesis. Succinyl coenzyme A is a central intermediate in the synthesis of porphyrin ring of heme groups which serve as oxygen carriers in hemoglobin and myoglobin, and electron carriers in case of cytochromes. And the citrate produced in some organisms is used commercially for a variety of purposes. Now the regulation of TCA cycle. The regulation of TCA cycle is largely determined by substrate availability and product inhibition. NADH, a product of all dehydrogenases in the TCA cycle with the exception of succinate dehydrogenase, inhibits pyruvate dehydrogenase, isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase and also citrate synthase. Acetyl coenzyme A inhibits pyruvate dehydrogenase while the succinyl coenzyme A inhibits alpha-ketoglutarate dehydrogenase and citrate synthase. It has been shown that calcium is used as a regulator. It activates pyruvate dehydrogenase, isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase. This increases the reaction rate of many of the steps in the cycle and therefore increases the flux throughout the pathway. Low levels of ATP stimulate the formation of acetyl coenzyme A for the TCA cycle, while high levels of ATP and NADH decreases the formation of acetyl coenzyme A and slow down the TCA cycle. Overall reaction of TCA cycle. The reaction is as follows. Acetyl coenzyme A combines with thrice NAD positive plus FAD plus GDP plus thrice water and gives twice carbon dioxide, thrice NADH, FADH2 and GTP in case of animals and ATP in case of plants plus water. Although the citric acid cycle directly generates only one ATP, plants and GTP in animals per turn in the conversion of succinyl coenzyme A to succinate. The 4 oxidation steps in the cycle provide a large flow of electrons into the respiratory chain via NADH and FADH2 and thus leads to the formation of a large number of ATP molecules during the oxidative phosphorylation. The energy yield from the production of 2 molecules of pyruvate from 1 molecule of glucose in glycolysis is 2 ATP and 2 NADH. In oxidative phosphorylation, passage of 2 electrons from NADH to oxygen derives the formation of about 2.5 ATP and passage of 2 electrons from FADH to oxygen yields about 1.5 ATP. This stoichiometry allows us to calculate the overall yield of ATP from the complete oxidation of glucose. When both pyruvate molecules are oxidized to 6 carbon dioxide molecules via the pyruvate dehydrogenase complex, the citric acid cycle, and the electrons are transferred to oxygen via the oxidative phosphorylation, as many as 32 ATP molecules are obtained per glucose molecule. In plants, the TCA cycle has unique features. The TCA cycle reactions are not all identical to those carried out by animal mitochondria. For example, the step catalyzed by succinyl coenzyme synthetase produces GTP in case of animals and ATP in case of plants. In animals, mitochondria, ATP is formed in a second enzyme reaction that involves the transfer of phosphate from GTP to ADP. Another feature of TCA cycle that is generally unique to plants is the significant activity of NAD+ malic enzyme, which has been observed in all plant mitochondria analyzed to date. This enzyme catalyzes the oxidative decarboxylation of malate. This is the reaction catalyzed by this enzyme. Malate plus NAD+ gives pyruvate plus carbon dioxide and NADH. The presence of NAD+ malic enzyme enables plant mitochondria to operate an alternative pathway for the metabolism of PEP, phosphoenol pyruvate, derived from glycolysis. Malate can be synthesized from phosphoenol pyruvate in the cytosol via the enzyme PEP carboxylase and malate dehydrogenase. The malate is then readily transported into the mitochondrial matrix by a decarboxylate transporter on the inner membrane that catalyzes the electroneutral exchange of malate and inorganic phosphate. In the matrix, NAD+ malic enzyme promotes the metabolism of malate by oxidizing it to pyruvate, which is then oxidized via TCA cycle. The presence of NAD+ malic enzyme thus allows the complete oxidation of organic acids, for example, malate, citrate, 2-oxidoglutarate, in the absence of normal TCA cycle substrate, the pyruvate. The presence of alternative pathway for the oxidation of malate is consistent with the observation that many plants, in addition to those that carry out crustacean acid metabolism, store significant levels of malate in their central vacuole. With this, we will conclude the topic Krebs cycle. Thank you for watching.